Okay, I'm here to introduce Dr. Kathleen Walker. She's an entomologist and associate professor at the University of Arizona. Dr. Walker studies the ecology of arthropod vectors of human diseases, particularly the invasive mosquito species Aedes aegypti, the primary worldwide vector of arboviruses such as dengue, Zika, and yellow fever. She explores the environmental and human factors that affect this mosquito's abundance and longevity in an effort to understand why arbovirus transmission occurs in some communities but not in others. She's collaborating with the Centers for Disease Control and Prevention and the Maricopa County Environmental Services Vector Control Division to assess and improve current integrated mosquito management methods. A new project in the Walker Lab involves the brown dog tick, Rhipicephalus sanguineus, which is a vector of Rocky-mounted spotted fever, but in only a few places, including Arizona and their neighbor state of Sonora, Mexico. The Walker Lab is studying the genetics of this tick to find out why it transmits the RMSF pathogen there, but not in other regions. They're also working with RMSF-affected rural communities to develop a sustainable tick surveillance tool. But I believe today she is here to speak to us about Aedes aegypti. Go ahead, Dr. Walker.
Thanks, Kristen. Yeah, today I'm going to focus on mosquitoes, although I, like many of you, I have learned to love ticks, or at least appreciate them. So today I want to talk about the work that we're trying to wrap up. The COVID kind of got in the way of things in Maricopa County in collaboration with the Maricopa Environmental Services Vector Control Division. So let me just share my slides. Everybody see them?
Looks good.
Okay. And I never know what to do with this. So I think I changed the title a little bit to really focus more broadly on the different aspects of this study. So it's really an operational study. It's looking at how real world vector control activities impact the target and sometimes the kind of the non-target vector species of concern. And it's funded by the CDC, and it's really a part of the effort to make sure that we are prepared in the event of domestic Zika transmission. And there's many, many people involved, both at the U of A and at Maricopa Vector Control. But let's just get started. Let me tell you what I'm planning to talk about today. So first, just a little bit more about the arbovirus transmission risks in the Arizona-Sonora region. And then the different pieces of this operational research project. We looked a lot at the effects of ULV adulticiding, which is standard operating tool for mosquito vector control. We also look at larval controls in a particular environment. We also, while this wasn't originally part of the plan, we also did some pilot study of the into-care audicidal traps. And we did some work on community engagement and the involvement of and perceptions and knowledge of participants in our study. And this is the U of A part of the team at the bottom, me, then Don Gouge, Mike Reilly, and Casey Ernst. The first three of us are entomologists, and Casey Ernst is an epidemiologist all at the University of Arizona. So first, I wanted to take you back. Remember the Zika outbreak in 2016? Honestly, it almost seems kind of quaint now that we are dealing with COVID, but it was a big deal. There was a lot of concern. There was a lot of infections and a lot of suffering. And that is really what propelled this study. I think it's also part of what helped found the PacVec, isn't it? I think. Anyway, so that's the starting place for this study. And as you know, at the time, there was a general consensus that the Zika was a big risk for Central American, Caribbean countries, but there was a thought that the continental US was less vulnerable due to our ability to control the vector. And we found out when local transmission happened in Florida that actually controlling the vector was not that easy, and it took a lot of effort. And so that really created this concern that in the event of another continental US outbreak of Zika or another arbovirus vectored by Aedes aegypti or Aedes albopictus, are we really prepared? So there's a good reason to study arbovirus transmission, or the risk of arbovirus transmission is fairly significant in the Arizona, Sonora border region. This is one of the only parts of the US where US states are actually touching a neighboring state that has active transmission of Zika, of dengue. And we know that the virus, the vector is well established throughout the Arizona Sonora region. There is transmission in central and southern Sonora of these arboviruses, less so along the actual border itself, although there have been some cases of local transmission of both dengue and Zika right in the border area. You can see on the map on the right. Some previous research that my colleagues and I have been trying to figure out why there is arbovirus transmission by Aedes aegypti farther south, but not along the border and not north of the US-Mexico border in Arizona. And there's clearly some climate factors. There are probably some living factors, but the risk still remains that here you have a lot of human communities with a lot of contact back and forth between arbovirus transmission areas and arbovirus-free areas. There's a lot of vector, and so at some point, presumably, a sick person can go home to Phoenix, be bitten by Aedes aegypti in their yard, and then you have the potential for a local outbreak. So that is the reason for doing this study here. And in fact, if you look at Arizona alone, so all of those red dots are places where Aedes aegypti has been found using different health department or vector control agency trapping. And then the shading of the different counties are places where there have been actual travel-related cases of Zika. So you can see you have the ingredients potentially for an outbreak. You have lots of vector and you have infected people. And particularly Maricopa County, more than half the state of Arizona lives in Maricopa County. There is lots of Aedes aegypti, and there is a fairly consistent introduction of Zika, of dengue, of chikungunya viruses due to people traveling and then coming home with the virus. So that's the big question. Okay, are we ready, or are we already potentially doing activities that are preventing an outbreak, or in the event of an outbreak, would we be prepared? That is really what is driving this research. So as you all know, the CDC has very clear recommendations for integrated mosquito management, including surveillance, reduction of habitat, actual control measures targeting the immatures, control measures targeting the adults, and then monitoring the effectiveness of the control program. And a perennial problem for vector control districts, and all of you in vector control districts can jump in on this, is it is so much work doing steps 1 to 4 that it is hard sometimes to do as much of step 5, the monitoring of the effectiveness, as you would like, because just conducting all the other activities pretty much takes up all of the human and time resources. So we made a partnership at the U of A with Maricopa County because Maricopa County has a really well-developed integrated mosquito management program, and it's enormous in its scale. And in fact, I think these data are old, so that involves weekly surveillance of, now it's over 800 CO2 baited traps that are run all year round on a weekly basis throughout Maricopa County. And then at least the Culex mosquitoes that are collected are tested for St. Louis encephalitis and West Nile virus. So it's a very intensive surveillance effort. Then that surveillance effort informs controls. So there's kind of proactive larviciding of public drainage features in neighborhoods that have a history of high mosquito activity. And then in response to the surveillance, there are different thresholds for either truck-mounted or handheld ULV pyrethroid adulticide applications. And sorry, I always have trouble with the bar of people in Zoom. There we go. There's some exceedances based on the number of mosquitoes of different species in a trap. And then, of course, if there is a mosquito pool that tests positive. Both of those are triggers for a ULV pyrethroid application. And the way that the county is divided is each of these CO2 baited traps is representative of a 1 square mile block. So if there is a trigger based on the surveillance, then that 1 square mile block will be treated with the ULV pyrethroid spray. And I was going to say, this is not a formal talk, so if people have any questions or comments, please just jump right in. So this is the partnership. Here's some of our partners down in the photo below. And there were really two foci of this research project. One was looking at the truck mounted, or as it also turned out, there were some handheld pyrethroid applications. So what are the impacts of those pyrethroid applications on the Aedes aegypti vector population? And second of all, if you actually conducted larviciding targeting these public drainage features, what are the impacts of that larviciding on the Aedes aegypti population? So the way that we defined the impacts were for the adulticiding, it was the impacts on mosquito density. That's very self-evident. Are there fewer mosquitoes after you spray? But the second question that we wanted to look at is the mosquito age structure changed? Aedes aegypti has a very continuous population, so it sometimes is possible that you could spray an area and kill the mosquito adults that are contacting the spray, but there are so many larval habitats nearby that you very quickly replenish the mosquito densities with younger mosquitoes. The interesting thing is, from a disease perspective, a young mosquito is not the same as an old mosquito. So that's why we wanted to look at the age structure. And then the endpoint for the larviciding was really focusing on the presence of viable mosquito larvae in these treated drainage structures. And this was funded by the Centers for Disease Control and Prevention. Their Zika BAA, I forget what BAA stands for. And the duration of the project was from April 2017, basically to the present. So this is the setup. So we could not work across all of Maricopa County. It's enormous, and I still am amazed at how well vector control is able to cover such an enormous area in a very diverse land use area. You have everything from very, very dense cities to suburbs to agriculture. So we focused on a little corner of Maricopa County, the southeastern corner, which has two largely residential towns in it. Gilbert, which is represented by the pink, and Chandler, which is the light green. And then there's also a little unincorporated bit sprinkled all across it. And this area was important because, first of all, it's highly residential, and it has a very well-established Aedes aegypti populations within it. There's parts of the county where it's hard to find aegypti, but this was one of the hot spots historically. So as I said before, Maricopa County has a very, very well-developed surveillance system where in each of these little, I don't know if you can see it clearly, but each of these little blocks on the map represents a mile square. It's got nice grids. And in each of those blocks, at somewhere in each of those blocks, Maricopa County Vector Control set up a CO2 trap. And that's what those little numbers, those little black numbers are. So what we did is we worked with Maricopa Vector Control to look at the histories of Aedes aegypti presence, and we selected 10 1-mile blocks in each of the 2 cities, so a total of 20 1-mile square blocks. We also paired the blocks, that's what these red rectangular outlines are, blocks that had similar residential features, similar densities of population, and that's more for the larvicide study that I'll talk about later. Then in each of these 1-mile blocks, we put up 3 BG sentinel traps. And they were basically all at residences. And we also did some work looking at dry wells, but I'll talk about that later. So let's just focus on the adulticiding. So, what I just described is we had these 20 blocks, and in each block, for each year, we would sample Aedes aegypti adults in the sentinel traps weekly. We started in July, which is the beginning of the rainy season, when the Aedes aegypti population starts to rise, although there is activity earlier, and we would go through October. And here are 2 of our entomologists, Lucy Lee and Shaq Nair, collecting the Aedes aegypti and other mosquitoes from the BG traps and then sorting them. I'm sure it's a very familiar activity for everyone. And here is a general trends that we saw across the 3 years. So the average Aedes aegypti, and this is males and females combined per trap per night. 2017 is in blue, 2018 is in orange, and 2019 is in gray. And you can see that you know, when we start in July, it was pretty much usually the beginning of the season. The trap counts usually increased, although in 2019 we had kind of an early rainy season. And you can see it goes up and down. Some years a little different. 2018 was definitely a wetter year and a more abundant mosquito year. We also had these late season hurricanes in October, so we got this huge peak at the end of 2018, which was very odd. But you get this general seasonality to it. Usually the bulk of mosquito activity is in August and September, but there is quite a significant tail in October. And in fact, if we trapped into November, we probably would have continued to trap some mosquitoes. So that's our sampling. And now the operational part of this, the study of the effects of ULV adulticiding, are special because it was not the University of Arizona that did the spraying or decided when to spray. And we didn't have it set up where, you know, these 10 1-mile blocks are treatments and we're just going to spray them whenever, and the others are control. Instead, what we were basically doing is, not literally, but figuratively following behind the Maricopa County Vector Control spray wagons, spray trucks. When they had to spray one of the blocks because of a surveillance-based trigger, either the exceedance in the number of a certain species of mosquito in their CO2 traps or virus positive mosquitoes, then they would inform us so that we could increase our surveillance right after the spray event. And we would actually, during that first few days, trap usually 2 additional times just to get a really close indicator of how the mosquito population changed immediately after the ULV adulticide application. So in summary, across the 3 years, we had 16 spray events in 2017 during the July to October study period, and it was all targeting July populations. either because of exceedances or because of virus-positive mosquito pools. And all of the treatments were using the pyrethroid formulation duet, which is sumetrin and pyrrolitrin combined. In 2018, as I said, it was a wetter year, so we had more surveillance triggers for ULV applications. And in fact, 3 of them were actually exceedances of Aedes aegypti, so we were very excited. And then the others were for Culex-related exceedances or virus-positive pools. And then in 2019, again, it was a less rainy year, so we had fewer insecticide applications, and they were all either the duet product or another pyrethroid product, Permanone, and that was targeting exclusively the Culex. So I want to say straight up, most of the spraying that we were measuring the impacts of was not actually targeting Aedes aegypti, and that is part of the nature of operational research. As it happened in these 3 years, the number of times that Aedes aegypti exceeded their threshold was fairly low in the 20 study blocks in which we were conducting our research. On the other hand, one of the things that we really wanted to know is how are all of these West Nile virus related control measures impacting Aedes aegypti? In the event of an Aedes aegypti vectored arbovirus outbreak, can we just use these established techniques that have been developed through West Nile virus? Can we apply them to the control of Aedes aegypti? So I'm going to show you, this is the 2018 data first, because it was the most robust year, and we actually had some Aedes aegypti targeted insecticide applications. And basically what you can see in this box plot is, can you all see the legend, or am I covering it up with people? The first 3 of these boxes are related to Aedes aegypti density as measured by our BG sentinel traps. Now, we also caught Culex quinclofasciatus in our BG sentinel traps, so that is what these last 3 boxes are about. And one thing I want to point out is that we were very careful in our research to use a totally independent sampling from the sampling method used by Maricopa Vector Control, because if we were both using the same sampling, there's a lack of independence. So we were, we had our own separate BG traps. And anyway, looking at the effects, this is basically the post-spray abundance of Aedes aegypti minus the pre-spray abundance in these first 3 boxes, and they have to do with different time intervals. Blue is 1 week before versus 1 week after, orange is 2 weeks before versus 2 weeks after, and then gray is 1 week before minus, versus 2 weeks after. We were trying all the different combinations to look for an effect. And as you can see, when the box is pretty much on the 0 line, there is no effect. And the statistical analysis found no effect. On the bright side, when you look at Culex quinclofasciatus populations, there actually was a statistically significant decrease post-spray, which is a good indication that they are getting population reduction, at least for a short time, with their sprays, but on Culex, not on Aedes aegypti. And then just looking across, combining all 3 years. Oops, sorry, anytime I'm trying to move this box, things go crazy over here. And then we combined all 3 of the years. We did a multiple regression first to determine whether there was an effective year, month, block, or the type of insecticide used on the change in Aedes aegypti numbers before and after the spray. There was no significant effect, so then we just went to a paired t-test of Aedes aegypti density before and after sprays using different intervals. For before, 1 week before, 2 weeks before, and then after, 4 days after, 1 week after, 2 weeks after, no effect. So I think that we can say pretty fairly to the question, do the ULV sprays reduce Aedes aegypti numbers? And here's a very famous picture. I think Smith took this one of the enormous pile of mosquitoes caught in a single night in a single CO2 trap in 2016. You sometimes have these sort of blooms of Aedes aegypti that were caught in the traps. So overall, in general, do the ULV sprays as they are currently practiced reduce Aedes aegypti numbers? And the answer is no. Which is disappointing, but it's not really surprising. This, I think, is the sense that certainly that Maricopa vector control described beforehand, and I don't know if other vector control districts have the same experience, that the ULV sprays primarily targeting West Nile virus vectors really aren't doing anything to the Aedes aegypti, at least the products that are being used here. But there's another part to the question that we really wanted to examine, because the fact is, here's Maricopa County sitting there with mosquitoes and with people arriving with virus, and yet there has not been any documented local transmission. Is it possible that this well-developed spray program that is primarily suppressing West Nile virus vectors is also suppressing Aedes aegypti, but not in a simple numbers game sort of way? We know that arboviruses typically need several days to develop inside the mosquito. That's the extrinsic incubation period, unless there's significant vertical transmission from mother to offspring. But assuming that there's not, the virus needs time to develop inside the mosquito. So even shortening the mosquito's lifespan by a few days has the potential to disrupt transmission. So the question is, okay, so the mosquitoes aren't less abundant, but are they possibly younger after a ULV spray? It's the mosquito passing the virus on. So that leads us to the interesting question is, how do you figure out how old a female mosquito is? We know they don't live very long. There are a number of different techniques. One of the oldest is actually ovary tracheation to assess parity, whether the mosquito has blood fed and produced at least one batch of eggs. Dr. Mike Reilly up here and his technician Teresa Joy have also developed another method using the expression of a particular gene, SCP1, that is upregulated at the time of the mosquito female's emergence from her pupal case, and then declines in a fairly predictable manner whether or not she blood feeds. And so this, by measuring the quantity of gene expression, it is possible to estimate, fairly roughly the number of days since eclosion that mosquito has lived. So we have these two different age grading techniques. They both have their caveats, but Mike really wanted to investigate a third technique, which is near-infrared spectrophotometry. So I briefly described the gene expression technique. Then another technique, which I'm sure many people are familiar with, is actually ovary tracheation. Here's the mosquito ovary. And before she has ever blood fed, her trachea are all coiled up. And after she has blood fed and produced at least one batch of eggs, the trachea uncoil, of course, because they're going to be involved in gas exchange for the developing eggs. And those uncoiled trachea remain uncoiled for the rest of her life. So it doesn't tell you, very far out into her lifespan as an adult. Typically, they achieve parity in a few days, but at least you can divide the nulliparous, which haven't blood fed at all, from the paris. And this is Teresa Joy, who is the one who has dissected I don't know how many ovaries in the process of this study. And the nice thing about parity analysis is it's very definite. If you have an experienced person doing the dissections, the disadvantage, of course, is it can really only look at that early stage of adult life, typically. So So with the near-infrared spectrophotometry, it's based on the idea of looking at the cuticular hydrocarbons in the exoskeleton of the mosquito, which is actually an older technique, but it was very difficult to do. And so this is actually using electromagnetic waves that are going to create spectra based on how, I guess, they're bouncing off of the different hydrogen to oxygen or sulfur or nitrogen or carbon bonds on the outer surface of the mosquito. And the method is you scan the head and thorax of the adult mosquitoes at a very close distance, and then you compare the reflectance spectrum that you get from that mosquito with a model of, based on hundreds of mosquitoes of known age, the spectrum that you get from them. And that's the technique that Mike Whaley's lab has really been working on. And it has great advantages. It's very, very quick. Once you've invested in the initial equipment, it's not really expensive, whereas the gene expression, the qPCR, is very time consuming and expensive. And another advantage is you don't have to smush up the mosquito to do this, so you can keep it for other analyses. So this is the scanning technique, and this is kind of a model of the different spectra that you could get, the blue being a very young adult, and then the red being kind of 9 days, middle-aged, and 18, and the black being geriatric mosquito. And I'm not going to go into all of his work on developing the model. This is his thing, but I just wanted to describe that this work has been done, and here's actually looking at just one of the models. This was based on mosquitoes of known age raised under semi-field conditions, because he quickly discovered that mosquitoes raised in a lab don't have the same kind of weathering of their cuticular hydrocarbons, and so you get a very different model. And you can see, if you look really close, you can see that the predicted NIRS age is not exactly the same as the actual age, but it's pretty good. Anyway, so these are the 3 different age grading techniques that we had available to work with. But we realized very quickly that it's one thing to set up traps to measure density, but trying to measure age of that same population is really tricky in the case of Aedes aegypti, where you don't get hundreds of mosquitoes per trap per night. You get a few. And even though we had 3 different traps from each treatment area to pool together, it still was hard to get large sample sizes. And of course, if you're trying to estimate age of a whole population based on a few individuals, there's a tremendous room for error. This is just some preliminary data using the SCP-1 analysis in the first year, 2017. And these are 5 different blocks that were sprayed. So these are 5 different spray events. And you can see the blue is the average age before the treatment, and the orange is the average age after. And you don't need statistics to know that this is not statistically significant. On the other hand, if you can see the ends, these are very small numbers of mosquitoes. So it's really not valid to say, make big conclusions when you have small numbers, but it's hard to get large numbers. So this is a real sticking point of this study. 2018 was the year that we had the most mosquitoes, and here are a bunch of different spray events. They're listed by block number, but you can see some blocks were sprayed more than once. And then the orange is the mean age representative before the spray. The gray is after the spray. This is using the SCP-1 age grading. You do see in a number of the blocks a decrease in the average age after the spray, but in a couple of the blocks you see an increase. And overall, there was no statistically significant relationship before and after spray. Then this is looking at parity, so doing the ovary analyses and looking at before and after the spray event. Again, you would expect, if the spray event was killing the older mosquitoes, you would expect fewer mosquitoes to be paris after a spray event. We didn't see any kind of statistically significant trend in that direction. And then this is using the NIRS method, The one advantage that we had with NIRS is that we could use mosquitoes that had been caught dead in the traps, as opposed to the other two methods, which require live caught females. So we were able to have slightly bigger sample sizes. But again, we did not have a statistically significant difference in the average age before and after spraying based on the NIRS age grading. So there's clearly limitations of the age grading techniques, no matter what we use. And a big issue is the lack of really big numbers. So we're still looking into different ways to do the analyses and different ways to pool the data so that we can get more robust numbers to do these pre and post spray comparisons. But based on our analyses so far, there is not a significant decrease in mosquito age after the ULV spray events. So that was discouraging. Now we go to focus on the juvenile Aedes aegypti and the issue of operational control methods to control Aedes aegypti larvae. So again, we go back to the study design. Here's our 20 plots, all the things that are outlined in the red. And each of the 1 mile blocks is actually in a pair. So you can see over here, there's 694 over on the left, and below it is 850. Those numbers, again, are the trap numbers that the Maricopa Vector Control used. And so one of those blocks we designated as our larval control block, and on the other, it was our untreated. So one of those are larval treatment blocks, sorry, and the other is the control block. And in the first year, our plan was to have each treated with BTI product in all of the visible public drainage features. So as you all know, Aedes aegypti is a real bear to control at the larval stage because it has so many cryptic, temporary, and small water containers that it uses as larval habitat. Most of them are in people's private property, so they're not easily accessed. So some work in Australia and also in Central and South America, looking at public drainage features as important Aedes aegypti habitat and much more accessible for larval-based controls. So what we did is go into these same 20 1 mile blocks in the towns of Gilbert and Chandler. And in each 1 mile block, we identified 5 drainage features. And what we really focused on, I learned way more about public drainage features than I ever thought I would know, is what are called dry wells. So in a lot of parts of the urban Southwest, and actually in arid urban areas around the world, there's a trend towards using the land itself to absorb rainwater runoff. So instead of having all of the storm water go into a central drainage feature, each 1 mile block is designed in its drainage features to absorb its own water. So each one is its little island. And at the end of all of the roadside gutters and so forth, there is a sunken dry well. In fact, I think I have a picture of it here. It looks like this. And it's about 30 feet or 40 feet deep. It's designed to be porous so that all the water collects into these dry wells. And then if they are built properly and maintained properly, all the water should be absorbed into the surrounding substrate within 72 hours. So that's what we were focusing on, sort of the end point of these public stormwater drainage features. And so in each of the 1 mile blocks, we identified 5 storm drains, and those were going to be our sentinel storm drain dry wells that we would sample for the presence of larvae. The larvicide was applied not just to those dry wells, but across all of the visible drainage features prior to the start of the research. And so Then every 2 weeks during that 4 months of summer, graduate student Daniel Williamson would sample each of the 5 dry wells in each block, both in the treated blocks and in the untreated control blocks. And if you look at that dry well cover, and many of you who've done mosquito sampling will go, oh my God, how could you possibly sample that, especially given the fact that it's 40 feet deep. A lot of the classic dipper methods that just don't work. You can't get things through the grating, and anything that you drop into the water, the mosquitoes will run away from. So Daniel developed, I think, this very clever technique using an inline pump that would fit through the slats, reach all the way down to the water if there was any, and then he could suck up 2 to 3 liters and inspect the water for larvae. And so that worked really well. And I actually think that that might be a good tool for any kind of surveillance of these kind of cryptic drainage features, because as it turned out, they're very, very productive in terms of mosquito larvae. So just to explain how it works, so there are 5 dry wells that were sampled in each of the 20 districts, which makes 100. 50 of these dry wells were located in treated blocks where every drainage feature was treated with larvicide, and 50 dry wells were in control blocks. In 2017, the initial treatment was BTI granules, and it was the Maricopa vector control that applied them. So we were so naive when we started this. We thought that, of course, the larvae are going to be killed. It's going to be a question of whether enough of them are killed, or whether it's going to have a population effect. As it turned out, nothing was dying in these dry wells. And we found out a lot of different things. First of all, it was very hard to find dry wells. We had maps, but the Maricopa vector control personnel were going by eye, and a lot of the dry wells were simply invisible from the road. So some of them got missed. But even those that were treated, many of them still produced mosquito larvae, although none of them were Aedes aegypti. They were all Culex fasciatus. And the other thing that we discovered is that some dry wells work really well if they're well designed. I mean, none of them are supposed to be mosquito habitat, right? But so in the town of Gilbert, they had very well-designed dry wells, and in fact, it was very hard to find any that were flooded, and so there were obviously no mosquito larvae in dry wells. But in the city of Chandler, they had a tremendous problem with clogged dry wells, and so we found mosquito larvae in many of them. But we also found that, for example, if you look down here at these 5 red dots, this was a treated block, and all 5 of those dry wells were treated with larvicide, and you can the dark spots mean that there were larvae present. So clearly the BTI granules were not providing suppression. It's not surprising, granules don't last that long, but this was even 2 weeks after the application. So in 2018, we decided we're going to do this differently. We have learned from our mistakes. I'm sorry, I realize I'm taking too long. Well, I'll just try to wrap this up. So we actually used 180-day 4-star BTI briquettes that should last longer. The U of A team actually applied them themselves to all the different storm drain features, and we realized what an enormous job it was. And it was also, seemed to really upset the neighbors. We had the police called on us not once, but twice for putting briquettes down drainage features. And we had reduced the size of the study to just the 10 study blocks in the city of Chandler due to the drainage issues. Again, though, we're not finding success. So if you look at just the initial data, so these are the treated dry wells with water, and then here's the treated dry wells with larvae. The BTI briquettes were not killing the larvae. And we also discovered in this year, in 2018, we had both Culex and Aedes aegypti. We had Culex in most of the dry wells that contain larvae, but we also found a few with Aedes aegypti, and they tended to be these extremely clogged dry wells, like this one here. If you look really closely at the close-up, you can see larvae right up on the surface where the water is. So it did seem to indicate that Aedes aegypti was exploiting these habitats, at least sometimes. We tried everything. So we tried more BTI, we tried BTI and Alticid. Nothing killed the larvae. And this, and of course, with the Alticid, we brought larvae back to the lab to see if they would eclose. They did. So we really were very surprised by these results. We thought that these are established larvicidal products. Of course, they'll work. But I think what we realized is that these storm drain dry well features are really, really hard habitats to control. So finally, we had just a focus on just a small number of dry wells in 2019. We initially started out with 25, but several of them got repaired and stopped clogging. So we were down to 12, and we treated 4 of the 12 with BTI, 4 with Alticid, and 4 were controls. But instead of just throwing the briquettes in, we realized that given the incredibly deep nature of the dry wells and the huge fluctuation in water volume, not to mention all the sediments carried in, that the briquettes that sit at the bottom might not be able to release insecticide that would come in contact with the larvae. So Daniel developed this biodegradable little floating raft for the briquettes, which I thought was very clever. So it was big enough to not pop out through the grating, but small enough to be placed into the grating. And finally, we got some degree of success. And here I'm just going to show you the raw data because it's just such a small sample size. Here are the 4 dry wells that were treated with the Alticid, and at least in 2 of them, although larvae were collected, none of those larvae were able to eclose. Whereas the BTI treated wells really were not successful, even though we put multiple briquettes in. So we did finally manage to kill the larvae. It took us 3 years, which is not what we expected. But we did figure that out. But then there's the question. So Aedes aegypti did use these dry well sites on occasion, but it didn't seem like it was a very typical occurrence, that these were mostly Culex quincunfasciatus larvae that were exploiting these dry wells. So it led the question to where is Aedes aegypti, because we're getting plenty in the traps. So we did just a small backyard larval survey in 2018. And one thing that we learned, and I'm sure Maricopa Vector Control can back me up on this, is that the culture in different communities is very different. And when you're dealing with vector control, you have to take into account the culture. And one thing we learned about the suburban Maricopa County is that it's a culture that really values privacy. So knocking on people's doors and asking to look for mosquitoes in their backyard did not work. People would not open the doors. So we had to actually recruit participants in the backyard survey with flyers and then go back later. That, of course, tremendously skewed our sample towards homes where there was an actual mosquito problem, but at least it gave us a place to start. And of the 45 homes that we surveyed just outside, we did find Aedes aegypti immatures in 10 of them, and they were in the classic small, obscure habitats that Aedes aegypti is known for. The saucers under plants, ornamental features like fountains and bird baths. And I have to say, these are the cleanest yards I have ever seen in my life. These are affluent suburbs. They're all subdivisions built by single building companies. So there's tremendous uniformity and tremendous cleanliness and organization. So when we first walked into the yards, we're like, where are we even going to look? There's no clutter. But we did, in spite of the lack of clutter, manage to find primarily Aedes aegypti. So this suggests that, in fact, focusing on the public drainage features is probably not going to get at the bulk of the Aedes aegypti if we are trying to control that vector. It could be useful for quincunfasciatus. So that led us to try something that was not in the initial plan, But given the limited number of these classic cryptic breeding sites, we thought maybe this is a situation in which one of these autocidal traps, like the Into-Care trap, might work. So I don't know how many of you are familiar with Into-Care. I imagine most of you are. It's basically a trap that attracts the female adult, and then she picks up larvicide on her body when she visits the trap, which she then accidentally deposits in other breeding sites when she oviposits in them. And the insecticide is pyroproxyfen, which is a growth regulator, but there's also the fungal pathogen Bavaria bassiana to kill her eventually. I felt like that was unnecessary, but so we just did very small 2 neighborhoods in Chandler, and literally just a city block. This is an aerial view. The blue thumbtacks are the people who agreed to participate. The red are the people who never responded or told us no to our faces. And so we set up 2 traps per house. And the houses where they didn't cooperate, we just kind of tried to pack it in around there using some of the public spaces. But it was primarily this area and over here. And this is a neighborhood that had tremendous Aedes aegypti problems. We checked traps weekly, and we also changed the pyroproxyfen sachets. We put out ovitraps as sort of sentinel oviposition cups around the houses after 8 weeks to see whether we would get pyroproxyfen delivered to these sentinel traps. And we also looked at the bell counts for BG traps. And just really quickly, we were worried that the traps would just dry out in the hot, dry Maricopa weather. And they did. You had to replace the water every 2 weeks or even every week, but they did work. They were, by week 4, every single Into-Care trap had Aedes aegypti larvae in it. None of these larvae successfully closed, so the pyroproxyfen was working within the trap. But what we really wanted to know were these ovicup bioassays when we placed the cups around the neighborhood to see what would happen. And basically, we put them out for a 4-day period. We then checked to see whether they had any oviposition activity by Aedes aegypti, any eggs in them. And then we took the cups back to the lab with the remaining fluid in it and placed 10 young larvae from our own lab colony and saw whether they could develop. And we got really kind of, it's a pilot study, but we got really nice results from one of the neighborhoods. Literally none of the cups that had gotten eggs in them, therefore had been visited by an Aedes aegypti, none of them had any successful eclosion. Every single larva died. And the other neighborhood, some of them did successfully eclose, but the majority of cups did not. So you can't go, get too excited because it was such a small scale study. We had great plans to expand it in 2020. And of course, you all know what happened in 2020, so we were not able to. But this is still something I think that would be worth exploring. And then finally, we really wanted to know how the survey was, or how the study was affecting the people who chose to participate. This was an operational study in the field, in people's houses. The majority of the houses where we conducted the trapping chose to stay with the study for all 3 years, or at least 2 of the 3. And Casey Ernst and Don Goosh did some surveying of the participants to find out what they thought and what they learned from the project. And one of the things that was most interesting that we found consistent all years that none of them ever reported, or very few reported, mosquito problems to vector control. However, lots of them, especially in the 2nd and 3rd year, would call Dawn for advice. So it really connected, the project connected homeowners to us. Then the other things, as you might expect, a lot of people reported eliminating standing water. Some use pesticide outside and some use repellent. We saw similar results in 2018. Again, nobody wants to call vector control, but everybody wanted to call us. And then the final year we did, Casey and Dawn did a more in-depth study. And here's some interesting findings that I wanted to include. So one of them was, who do they feel should be responsible for vector control? And interestingly, the vast majority of those surveyed, it was more than half of the participants responded, identified the residents themselves as being responsible. So that was interesting. And then another interesting finding was asking people how much they would pay to have their mosquito numbers reduced, either through something that they bought or through a service. And again, this is an affluent suburb, but the vast majority expressed a willingness to pay. In all of these, 6% said they wouldn't pay, but that was one person, but everyone else said they would pay, and some of them quite substantial amounts of money. So this is obviously a biased sample, and that these were individuals who agreed to collaborate with us in this project, but still, I think that that's some interesting information. So in conclusion, the ULV sprays did not have an impact on Aedes aegypti. We're doing some resistance studies, and I know others of you were involved in them. There's clearly a high level of resistance. In terms of larval control, we found that these dry walls were really important mosquito habitat, but probably not so much for Aedes aegypti. We also found that they were challenging habitats in which to apply larvicide. And then we got some interesting results from the pilot study of in-situ care traps and the community participant reports. So I just want to acknowledge the incredible collaboration with everyone in Maricopa County Environmental Services Vector Control Division. And I really want to thank all of the resident collaborators in Gilbert and Chandler. Every week we would give each participant their weekly mosquito report, and people were really very appreciative of that information. I don't know what they did with it, but they still appreciate it. And of course, this research was funded by the CDC. So thank you.